Welcome to the incredible world of polar microbiology, where life finds a way to thrive in the most extreme environments on Earth. In the freezing temperatures of the Arctic and Antarctic, where conditions are harsh and unforgiving, tiny microorganisms are not only surviving but flourishing. These resilient organisms, known as polar microbes, have adapted to endure temperatures that plunge well below freezing, high salinity, and intense ultraviolet radiation. But how do they do it? The secrets of their survival lie in their incredible adaptations. Delve into the fascinating world of polar microbiology, where survival against all odds is a testament to the resilience of life. Join us on microbiaX Live with our featured speaker, Rantim Bhattacharjee, Ph.D. Fellow, National Centre for Polar and Ocean Research, India.
So let's start the meeting. Good morning to everybody who are attending this meeting and welcome to all. I'm Sajeetha, your host for the day, and I have with me today Mr. Rantim Bhattacharjee. He's a PhD scholar from National Center for Polar and Ocean Research. And we are glad to have you, Rantim, with us today. So before we get into the presentation, just a casual talk, like you could just introduce about yourself, like what made you get interested in polar research and what is so fascinating about polar microbiology, and like you can just give us some highlights about it.
Yeah, actually, from the childhood days, I was really interested about this polar region, but I didn't know that I have a knack for that. So once finished my schooling, then I went for biotechnology, and then I came to know about the fascinating stories about the microbes and their abilities to do, because microbes are ubiquitous. So after studying about this, then I came to know about this polar region and NCPOR as doing this. So I got interested and uh, fascinated about the abilities that is there for the microbes. Like they can survive anywhere. So it's
Yeah, that's
really
true.
interesting to see how they are doing that. So that's kind of fascinating to me. So I wanted to study those. That's why I'm here and I'm now embarking on this journey of PhD so that I
Yeah.
can learn more about this. That I
That's
can
good. That's good. Because polar microbes are not very common. Like most of us who take up microbiology end up with industrial or food or medical. That's a very common path people normally choose. But getting into the polar microbes and studying about organisms that are facing really the worst and harshest climate and how they are surviving and adapting. It's a very interesting field to ponder into. So let's get into your presentation.
Good morning to everyone. I'm Rantim. I'm a PhD fellow, as Sajeetha mentioned, at the National Center for Polar and Ocean Research in Goa. Today, I'm going to talk about the polar microbes and how they are surviving in the extreme polar environment. So, our topic is titled as Survival Against the Odds, Polar Microbes in the Extreme Environment. Now, let's talk about the Earth's poles. Yeah, we all know that North Pole, South Pole is there, but there is one third pole that is in between. So north is in the farther north, that is called the northern hemisphere, and then south, and in middle there is a third pole. Let's first talk about the North Pole. North Pole is situated at the farthest north, and in childhood we often refer to the North Pole as the home of Santa Claus. The Polaris or North Star appears stationary in the northern hemisphere, making it an excellent celestial navigator of the Arctic. Yes, it is also known as a Arctic Circle, and it is mainly composed of 8 different countries. The countries that made up the council includes Canada, Greenland, Iceland, Norway, Finland, Russia, and the United States of America. They formed an intergovernmental forum called the Arctic Circle, and they actually govern the Arctic research policies and recent climate change. It is also surrounded by the Arctic Ocean, or also refers to as the High Sea, and always covered in 2 to 3 meters of ice sheet, while the ocean depth sometimes exceeds up to 400 meters. Arctic experiences two types of climate. One is maritime climate and another one is continental. Maritime climate is experiencing cool, stormy weather in winters and summers averaging in 10 degrees centigrade in average. But in the interior part of the climate, where things get a little harsh because in winter the weather becomes so dry and the temperature drops around 40 degrees centigrade. And in summer it averages around 30 degrees. So it's a harsh one, harsh climate to be. Due to the axial tilt of the Earth, North Pole gets a sunset and a single sunrise every year. So yes, phenomenon that is called 24 midnight sun, or they get 24 hours of day and 24 hours of night. In the nights, especially in the colder nights, there is a beautiful phenomena that occurs, and it's a mesmerizing cosmic dance of the light that is happening. It is called the aurora borealis, and it's really beautiful. Or sometimes it is also known as the northern lights. It's a great sight to see. So that's all about the Arctic basic features, and then we can just move on to the South Pole. That is also known as the Antarctica. It is the southernmost point of the Earth, and Antarctica is the only continent that is surrounded by the Southern Ocean. The 360-degree area, which is the only continent, is made up of ice, and it is surrounded by a single ocean. Antarctica is much colder than the North Pole. It has an elevation and also has a 2,700-meter thick ice sheet, where the warmest temperature has been recorded minus 12.3 degrees, and the coldest has already been recorded is minus 89.2 degrees near the Vostok research station of Russia. Like the North Pole, the South Pole also experiences extreme variations, and it also observes a single night, a single sunset, and single sunrise. 24 hours of day and 24 hours of night is also there. It's just the opposite because in the southern hemisphere, when the summer is there, northern hemisphere experiences winter. So there are equinoxes like March and September that is marked by the sunset and sunrise time. Now the South Pole is constantly shifting with a ice drift about 10 meters annual. Due to this slow movement, the Earth's tectonic plates is responsible for this, and for the billions of years, the continent is drifting away. Here is also you can get to see the aurora. It is known as the aurora australis, and few people actually get to experience this type of thing. They went to visit the Antarctica. They are mostly scientists and explorers. So once we had a meeting here where the people from the first Antarctic expeditions came and they shared their experiences and they interacted about this. So it was really nice, and we get to know a lot that how harsh the temperatures could be, how tough to survive in the Antarctic. Now I'm at the third pole. Yes, we have a one polar region. It is the Himalayas. The Himalayas are considered as the third pole because it has vast glaciers, which holds the third largest amount of fresh water after the Arctic and the Antarctic. Now the glaciers in the Himalayas are really crucial because it contributes to the its meltwater actually contributes to the 10 major reservations in Asia, including the Indus and the Ganges region. With combination of them also, they ensure water supply to the billions of people. Now, the temperature here varies in summer, averages from 15 to 30 degrees centigrade, while in higher altitude, the snow is prominent. Temperatures often hover around 0 to 15 degrees. In summer, in summer, and it drops well below to minus 30 to minus 30 degrees in winter. Himalayas are beautiful, so you can know that yes, it is also considered as the third pole because the climatic conditions are almost similar to the Arctic and Antarctic. Now I'm going to discuss about the Indian Polar Research Station. This is something we all should know and we should discuss. And I saw this opportunity and I am actually talking about it. So, India has established three research stations in the Antarctica, which are Dakshin Gangotri, Maitreyi, and Bharti, of which Dakshin Gangotri was the first one to be established. The first Indian scientific expedition happened in 1981, and this research station was founded in 1983. But due to severe snow and snowfall, snow accumulation, this research station was decommissioned in 1990. Dakshin Gangotri is a part of Indian research stations in Antarctica and it has helped carry out a lot of good research activities in the southern continent. Dakshin Gangotri had an automatic weather recording station which was fully powered by solar energy, and it also helped in observations of physical oceanography and chemical oceanography, which includes the knowledge to enhance our understanding of the climatic pattern which can affect the monsoon and the weather system of India. Also, there has been a lot of new species discovery happen, and you can check it out. You can just search for it. I have not mentioned that. Now, the second one is the Maitri, that is India's second base station in Antarctica. It is established in 1989 and still operating. This modern research station has different disciplinary studies such as earth sciences, glaciology, atmospheric sciences, human physiology, medicine, meteorology, and others. People can actually apply for these expeditions and they can go on an expedition to explore the Antarctic region. And if you want to study, you can also apply through. Now, the third base station, scientific base station is Bharti, which was established in 2012. It's fairly recent. It includes a fuel farm and a station for seawater pump, and it also has some market. Bharti mainly focuses their research on plate tectonics and geological structures, and the astronomical and remote sensing is also studies are done. It is done by ISRO and NRSC along with NCPOR. India also has a base station in North Pole that is named as Himadri. Himadri is the first permanent Arctic base station. It is situated in the Ny-Ålesund, Spitsbergen and Svalbard in Norway. For Himadri, it has primary research goals that encompasses aerosol radiation, space weather, food web dynamics, microbial communities, glaciers, sedimentology, and carbon cycling. This research base also focuses on the governance and policy of the Arctic research. The priority to the Indian research includes genetics, glaciology, geology, atmospheric pollution, and space weather, among other things. Next one is Indian research stations, which I would like to mention about the Mooding Point. It is the IND-ARC, is India's first underwater mood observatory in Arctic region. It is deployed in 2014 at the Kongsfjorden in Svalbard, that is just in between the Norway and the North Pole. This observatory aims to study the Arctic climate and its impact on the monsoon. There is another collaborative lab that is known as Udvadet Atmospheric Laboratory, which is actually managed by Norwegian Polar Institute and Kingston. It is it was established in 2019 to facilitate easier access to the rare users like CNR, NCPOR, NIPR, and KOPR. These are the renowned polar research institutes in the world. This facility currently houses instruments for aerosol sampling among with other research projects. Now in Himalayas, Himans Himans is another research station that is present in the Himachal Pradesh. It is located at the Sukri Dhaka in the Chandra Basin of Lahore Spiti Valley, and it has an elevation of around 4,080 meters above sea level. It was inaugurated in 2016, and at that 2016, this research station was closed due to heavy snowfall. But after that, it has been continuously running throughout the year. The laboratory equipped with advanced scientific instruments including a steam drill, ground penetration radar, GPS, snow ice scorer, snow tracker, water level recorder, automatic weather station and radio. These tools are actually very important in zoological research and glacier dynamics has an impact in interaction with the Chandratal glacier and its glacier. So you can see that there are three huts. Two of them is for residents and one is the laboratory. Now I'm going to talk about some interesting features about the North Pole or the poles. Now the polar regions are actually characterized by their extensive floating sea ice. This is a phenomenon you can only get in the polar region. The Arctic and Antarctic ice sheets cover the whole ocean, northern and the southern ocean. These areas receive less direct solar radiation due to the sun's oblique angle and Earth's axial tilt, which actually contributes to the colder winters compared to other regions. The presence of snow causes an albedo effect, which reflects the sunlight and contributes to their cold weather. Now, the interesting fact is these polar ice caps actually holds the most fresh water in the world. See, the Earth has 70% of water, but only 3% of Earth's water is fresh or usable. Now, of this 3%, only two-thirds of that is locked in the glaciers and the ice caps. Of this, 90% is present in the Antarctic ice sheet and about 9% is present in the Greenland. So, I am a is part of the institute that is known as the India's gateway to the polar region. That is National Center for the Polar and Ocean Research. Formerly, it is known as National Center for Antarctic and Ocean Research. NCPOR is an autonomous research and development institute which is based in Vasco da Gama. NCPOR actively participates in global experiments, hosts international conferences, and leads international committees focused on the polar and ocean science. NCPOR's mandate is conduct a wide range of scientific activities, including polar expeditions, oceanographic research, climate studies, and environmental monitoring. The responsibilities include collection and preservation of ice core samples from Antarctic, Arctic, and the Himalayas, managing and conducting operations of the research stations that are situated in the polar region, and maintaining the ORV, or Ocean Research Vessel Sagar Kanya, that has helped a lot of study in the Indian basin of Arabian Sea, Bay of Bengal, and the Southern Ocean also. Now I'm coming back to the topic that is microbes in the polar regions and how they are surviving and how they can endure the harsh environment. First, we can just see about the diversity or how the polar regions are composed of microbes. There is a good study that is how et al. Has done. They have collected samples from the polar regions as you can see in the map. The blue ones are the study points in the Arctic region and the red ones in the Antarctic region. So after metagenomic study, they came to know that and comparing with other databases like Tara Ocean databases, they get OTUs that are known as operational taxonomical units. So this operational taxonomical unit actually conducts about the unique species. So they have found around 7,520 unique species in the polar region. And in the right side, you can see the different composition of the bacteria that is made up of. You can see the group of bacteria that is majorly dominated by gamma proteobacteria, alpha proteobacteria, pseudomonas, and others. Now these bacteria are actually involved in carbon fixation, nitrogen cycling, silicon cycling, and liquid sugar synthesis. There are other studies too, which is done by Mallard and Pierce. And from the Arctic soils, they collected samples and they can here you can see the composition. Cyanobacteria, Actinomycetes, Chloroflexi, Firmicutes. These are the actinobacteria. So at the right, you can see this one actually represents the composition of bacteria from Arctic, Antarctic, and the Alpine. This is in the phylum portion, and this is from the class. So as you see in the class portion, the Arctic and Antarctic are much similar, so which states that these bacterial compositions are almost similar in polar region. Now for the Antarctic soils, you can see the dominating part of cyanobacteria that are dominating in the Antarctic soil. And there are a lot of genera that can contribute to their bacterial community. The changes, challenges, and the survival. The first challenge is the cold temperature. You always know that ice means cold, so microbes cannot survive in solid ice. But within the ice sheets and glaciers, some bacteria thrive in tiny liquid veins formed during the freezing temperature. Now, when the seawater freezes, the ice crystals actually reject the dissolved salt, microbes, and the water gets trapped into salty brine. This harsh environment is very difficult, makes very difficult the microbial life to thrive. But still, bacteria overcomes it. So this is the interesting part actually, how they can overcome. And another problem they have is bacteria is made up of mostly water. So when the ice freezes, it forms little ice crystals. These ice crystals can grow outward and pierce through the bacteria, eventually killing them. So as you can understand that this is a very difficult situation for the microbes to be in. So for survival in the cold temperature, microbes develop several strategies. First one is nutrient transport. With this nutrient transporters, they acquire nutrients from the slow moving environment and it helps them keep going. Nutrients are actually the ingredients for the cooking of the food, as you can see. Then again, another strategy is they deploy cold enzymes or cold active enzymes. See, in other enzymes, they need a certain warm temperature, like 37 degrees centigrade. They work best there. But these cold active enzymes can work at low temperature, which is really helpful for those bacteria. Another master stroke is producing antifreeze protein. This prevents the ice crystal formation in the cells and eventually damaging the cell. The fourth and other strategy is absorbing dissolved salts and sugars to lower the freezing temperature. The salt water or the sugar water, when it is mixed with the solution of water, it lowers the freezing temperature. The alpha amylase is a primary cold adaptive enzyme. And it is also secreted by Bacillus, Cryptococcus, Penicillium, and Pseudomonas. Where AFPs or antifreeze proteins are primarily produced by Cryobacterium, Mexophonia, Polaromonas, Pseudomonas, and Sebastopol. Adaptation also happens for the microbes in the cold temperature. These adaptive microbes are known as psychophiles or psychotrollins. They undergo numerous cellular biochemical modifications to mitigate their effects on low temperature stress. When the stress happens, they get to physiological adaptation, which contributes to the metabolic changes, and metabolic changes goes to enzymatic changes. Yeah, we can also see their gene expression changing, which eventually helps them in mitigating the temperature stress. Also, after years of study, it is found that general mechanisms include cold shock protein increasing, increased membrane fluidity, presence of compatible solutes, replace oxidative metabolism, structure adjustment of enzymes, then also contributes to their cold adaptive state. Other mechanism also there, that is secondary metabolic pathways, that lyophysolate pathway, and also they make changes to the cell wall also, like they secrete more lipid substances so that the fluidity maintains. And also EPS is a good example for survival in the cold temperature, that is the polymosaccharide. Now the next challenge is high UV exposure. Now ultraviolet rays are a type of electromagnetic radiation from the sun with shorter wavelength than the visible light. So if you see the electromagnetic spectrum, the VIBGYOR is like violet, yellow, red. So ultraviolet is shorter than that. Now large doses of UV rays can harm humans, animals, and proteins by causing disturbance to their immune system, cataracts, and eventually causing cancer. It is also harmful to the plants and any living thing that is there. So polar life is vulnerable to the UV radiation due to the cold climate and low sun, which actually get increased exposure in spring and further risk the melting of the protective snow of ice due to global warming. UV light also inactivates microorganisms by forming pyrimidine dimers in RNA and DNA, which can interfere with transcription and replication. Research has also examined how UV radiation enhances the toxicity of certain chemical compounds, particularly those linked to the oil spill for petroleum contamination. It is also called photo-enhanced toxicity. It is very harmful and can even kill certain specific species. So first is the cold temperature and second there is UV exposure. How they can survive this. Now some organisms also evolve strategies to protect themselves and repair the damage that has been done by the UV. But still they can be altered. Now there are certain examples or case studies I can see that carotenoid type of bacterial pigment usually shields cells from harmful UV radiation, which actually facilitates the bacterial survival in the polar region. Next is Chlamydomonas nivalis, produces a pinkish red pigment, protect themselves from intense UV radiation, giving the snow a slight watermelon shade. You can see in the bottom that is the watermelon snow, and it's really good to see. It's also reported that it has a written watermelon scent also. Rhodobacter species also adapted to the osmotic stress in the hypersaline environment, shows increased resistance to UV radiation, supported by the high levels of osmoprotectant related proteins that act as effective reactive oxygen as a species scavengers under UV exposure. Also, another strategy is they bury themselves in deep on the ice and snow sheet, which eventually the sunlight can't penetrate, but due to global warming, that is also happening. So, UV exposure usually leads to the generation of ROS or reactive oxygen species. Those are known as superoxide molecules. They can harm or damage DNA and proteins, fat, and it can also be linked to cellular dysfunction, aging, and diseases like cancer. ROS comes from places within our body, mitochondria during energy production, enzymes in cellular production, and the immune system fighting infection. Also, due to metabolic stresses, ROS can be generated. ROS can be of six forms: like oxygen, then peroxide ion, hydroxyl radical, and hydroxide. To combat this, exopolymeric saccharides or EPSs are recognized for their ability to shield microbial cells from ROS. And also, the compounds they produce that is also known as antioxidants. We have also heard about antioxidants, and it actually relieves in the oxidative stress produced by the ROs. Now, another problem they face is low water availability. So, when the ice freezes, the water becomes very less and it is very freezing, which can be detrimental to the cell. The microbes form a dormant spores or cysts that can withstand for prolonged time and it does not require any nutrients or water to survive. So that's how they combat the low water availability problem. Next up is the seasonal variability. The polar regions experiences extreme seasonal variability with prolonged night through periods of darkness in winter and continuous daylight in summer. 6 months they get summer, 6 months they get winter. All the time they are light and night. How they survive. The microbes also adapted to the seasonal changes, adjusting their metabolic activities and growth rates. Some of them also hibernate or remain dormant during the dark night and resume activity during the polar sun. The basic form is forming the dormant cells, the making the cells dormant and forming the spores and cysts. Second problem is nutrient availability. Because in freezing nights, where essential nutrients like nitrogen or phosphorus gets very restricted. This inhibits their growth and metabolism. So microbes utilize efficient nutrient acquisition mechanisms by producing extracellular enzymes to break down complex organic matter that is known as POM or particulate organic matter, and it is obtained from the DOM or dissolved organic matter. And last but not least, oxygen levels can vary being too high or too low in the polar environment, particularly in marine ecosystems where ice cover and microbial activity influence oxygen concentration. So some microbes turn to anaerobic conditions to adapt to the low oxygen or hypoxic situations, and some form symbiotic relationships with other organisms such as phytoplanktons and diatoms. They can get the oxygen they need because they also survive in those cloned environment by restricting their metabolic processes and their growth. Now, why we need to study the microbes? What are the usefulness of the microbes? Let's talk about the research and application. First up is the endophase protein. It's really interesting to me because endophase proteins can be used in various reasons. Like it can provide you the freeze tolerance that is required and the addition that is required for the microorganism to grow. It is also used in the freeze avoidance. See, when the water inside your body freezes, this endophase proteins actually protect you to not be killed or not get any damage from it. So this tolerance is actually getting from the AFPs that contributes for the plants and nematodes. They usually release them. There is one nematode that can survive up to minus 80 degrees centigrade. You can imagine the endocrine proteins are really important for this guy. Otherwise, they will just be killed. Another main function of AFPs are they provide a surface for the microbes to inhabit so that they can grow and get the light and oxygen required for their metabolic activity. And for the fish and insects, antiphase proteins we used for maintaining their fluidity. Antiphase proteins are also found applications in fertilizers, crop promotion, pest control, and improving freeze resistance in transgenic crops. It is now finding its own way in biomedicine and cryosurgery with enhanced effectiveness. AFPs are also used in food storage. It is also very useful to process and store the freezing damage in foods like meat and bone. Also enhances the texture and flavor, freshness, although the potential allergy risks are there. AFPs are pivotal in controlling ice crystals growth in refrigeration and influencing precipitation and cloud formation for climate management purposes. Second one is the cold adaptive enzyme or cold active enzyme. From the colder environment, you get the sacrifice of cyclotrophs. Now, these cyclotrophs actually survive by secreting cold adaptive enzymes. They help in reducing hydrophobic and electrostatic interaction, reduce hydrogen bonds in salt disulfide bridges, They provide a great surface for hydrophobicity, while substrate for pocket size. They also lower the arginine or lysine ratio that increases the glycine residue, and they also reduce the toluene content, increasing tolyl isomer. Now, this cold active enzymes has various applications. One of the most important is in the food industry, like dairy, meat, bakery, brewing, and juice and wine production. So I have actually mentioned some of the enzymes, that is lactase, protease, catalase, papain, glucose oxidase, and lactase, lipase, etc. These collective enzymes helps in degradation of phenolic compounds and aromatic amines at colder regions. It's also used in treatment with wastewater, with cold oils in cold regions, and treatment of xylene contaminated environments at low temperature. It is also used in iodination, dairy and bakery products, and also used in detergent and fabric industry. It has found usefulness in removing greases. Now, for medicinal purposes, cold active enzymes are used for thermolevels, ingredient synthesis, cosmetics, fragrances, pharmaceuticals, with applications including drug intermediates. There is one cold active enzyme that is known as lipase. It effectively removes oil from the wastewater and lactase that can degrade pollutants under mild conditions. Offer an eco-friendly and energy efficient alternative to harm chemicals in the detergent industry. It is also used in blood transfusion therapy, therapeutic agents, and beauty. So let's recapitulate what we have talked about so far. That first I mentioned that Earth has three poles, that is Arctic, Antarctic, and Himalayas. Then we came to know about those basic features, and same some features of the North Pole, South Pole, and the third pole. Then we learned a lot about the Indian research space stations like Lachlan 3, then my 3 RP that are situated in Antarctica, then Himadri in the Arctic, and Himans in the Himalayas. We also get to know about the extreme cold and high UV exposure and how the bacteria get survive in that, how they are adaptive, how their antiphys protein and enzymatic activities change their way of life, and how they are uh, adaptive to that particular condition. We also seen some examples of application of the antiphys proteins and active enzymes produced by the microbes in the solar. Do you have any questions? I think we have time to get some.
Thank you, Rantam. Thank you so much for helping us understand what these polar microbiology, importance of polar microbiology, and what these organisms are doing to survive in such harsh climate. It was very interesting to note how you had, structured the conversation, explaining about the polar, and then coming into the details of microbes, the challenges they face, and the strategies they use. One of the greatest and most important features of polar microbes is like antifreeze proteins. They have wide applications in biotechnology. So one of the important applications of using polar microbes is you can use it in biomimicry also. So I'd be discussing about biomimicry later part. So are there any questions for Rantan right now from attendees? Is there any question that you would like to ask him?
Microorganisms. Joy asked me, microorganisms changes in different layers of ice. Yeah, actually their community structure changes a lot. If you take the different layers of ice and study it metagenomically, you'll get to know that in deeper layers the pressure is much more higher. So you can see the dominance of barophytes that can withstand the pressure in there. And if you go up in the upper layers, you will get to see that the more UV tolerant bacteria are there. So there are certain programs where we can actually people from NCPOR actually go to the North Pole and drill inside and collect the ice cores. from that ice cores, we actually get to study that this type of bacteria can withstand this much pressure because the ice structure itself changes in the deeper layers. So yes, in different layers, we get to know the uh, temperature temporal cycles. Like these are paleoclimatic studies they do. They take the ice cores and kind of use it for IRMS, and they get the ratio of that. You can see also that different types of bacteria is also present, and it is mostly in the dormant spores because they form spores to survive in this half.
Are there any other questions for Rantim? Yeah, there's one question for you. Um, are only oligotrophic bacterial found in the stress condition? What kind of culture medias are used to isolate them? And another question is from Ritu. She has asked why we are interested in cold active enzymes like lipase and protease. Is it generally found in all organisms, and why do you prefer it from polar microbes for it?
Not only oligotrophic bacteria, actually. We can find different types of bacteria. Uh, mm, there are certain bacteria that can withstand the bacterial stress conditions, and culture media that depend upon which type, from what environment you are isolating. So, in marine environment, we actually use the marine agar, like Zobel marine agar we use, and also artificial seawater medium we also use to isolate those. And if you go to the soil part, then you can go for any like urea Bartini or nutrient agar. See, the cold active enzymes are very interesting because other organisms that produce in normal climate that requires a temperature in higher temperatures, like 30 above 30 degrees, they are required to get activated. But in cold active enzymes, the temperature does not require to go that high. They can activate within 10 degrees centigrade. So it is very easier and it is very useful to collect those and use them as a material for food industry and food preservative. That's why we prefer it for the polar micro. And this is just the two things we know. There are a lot of numerous studies, and these are the most significant I came across. And people are actually researching on it because you see, 95% of the total bacterial population is unknown to us. So there is a great black hole or black body is there, the dark matter we say. So this dark matter needs to be assessed, and more we came to know about this, the more possibilities there that we can learn about the other processes.
Are you able to see that, Rantam? There's a question in the chat.
Can't actually seen the last one.
She's asked, like, can you give any insight on how these psychrophyls can help in plant growth promotion or agriculture? Do they have any bioremediation potential?
Yeah, they have bioremediation potential. I can refer to you some excellent research works if you want. So that will elaborate more than myself because that is good because I really don't work on those particular things. So I just read them. So if you want to explore more, I can suggest you some research works of the paper.
Okay, so I think Niharika, your mic is on. I've allowed your mic. If you want, you can just talk in person, like directly on mic.
Thank you, thank you. Yeah, actually, uh, I really would like if you can give me any, any research work or any research studies based on this bioremediation potential or, the use on agriculture or crop production or something like that of
Yeah,
particularly this cycle.
actually, I have seen some reports that they are using the collaborative enzymes to make them grow in the frosty environment. So they are used as a cryoprotectant. So, and these Microphiles producing these collaborative enzymes are used in uh, the preservation of the food and everything. And they also have bioremediation potential. Like, yeah, I will give you those. If you can share me your email, I can send you some or give me
Yeah,
your
yeah, sure. Sure,
yeah.
I'm just writing it on the chat box. Another thing was like, is there only this culture dependent studies are conducting right now? I mean, the work you are doing, is it completely based on culture dependent studies, or culture independent studies are you're also conducting them side by side?
Okay, so we are focusing on both. See, not 100% of the bacteria, or just 1% of the bacteria is getting cultured. That is in the culturable form. So for unculturable forms, we tend to study for the metagenomic part, because we get to the samples and we collect the water or ice samples, whichever is collected, and we isolate the DNA or metagenomic DNA, the whole DNA of the sample, and then we subject it for different gene of interest. So that's how we came to know that okay, these genes are present in that particular set of samples or not. So different layers we take and then we go for that. Other is culture dependent studies. That is a bit difficult because not every bacteria grow on the lab. Since we
Right.
can get to a little bit of that. So, yeah, we are actually working on those.
So I'm writing my email ID. If you just can send me feedback on
Yeah,
those.
yeah, yeah. I will send you.
Okay, so that leaves us with the. Are there any more questions here? Okay, so let's just say thank you very much, Rantam, for sharing your insights about polar microbiology. It was really informative and very helpful, and I'm sure people would have enjoyed it. And thank you so much once again to all the participants who joined us. Thank you so much for sharing, and I hope people who are interested in microbial biomimicry will also be trying to look into the scope of polar microorganisms for biomimetic applications. So with that, I would like to end this webinar, and thanks to the participants once again. Bye to all.
MicrobiaX, microbe-inspired innovation.